एक बार चेक करे, कुछ बोल के देखे तो हम एक बार बाहर जाते हैं एक बार।
आवाज आ रही है?
हेलो, माइक इसमें देख, आ रही है ना उसमें, कुछ बोल के देखे। हेलो, साउंड ही नहीं आ रहा। नहीं आ रहा आवाज, नहीं आ रहा।
Contribution to various institutions. He has been an assistant professor in the Department of Genetics and Plant Breeding, College of Agriculture, Dharwad. He has been a visitor of the Department of Ecology, Evolution and Behavior, University of Minnesota. He has been a research associate in the Evolutionary and Organismal Biology Unit, Jawaharlal Nehru Center for Advanced Scientific Research, research scientist in Bangalore. He has been a visitor at Department of Biology, University of Regensburg, Germany. Sir has mentored several PhD students and has a number of research articles and papers to his credit. Professor Lalita Gupta is a professor in the Department of Zoology, Chaudhry Bansilal University, Bhiwani, Haryana. Ma'am has a PhD from the Banaras Hindu University, and from there on she has been a recipient of postdoctoral fellowship in France, postdoctoral fellowship Colorado State University. She has been a research fellow at the National Institute of Health, USA. She has been a recipient of several and of NIH Employee Special Performance and Achievement Award. I again welcome Professor Sakara and Professor Gupta to the session, and I would like them to take on the session. Thank you.
Thank you, Mamta. Our first speaker is Professor K. Raji, who is a senior professor at So I will not.
ये तो विजी है ना HDMI सर्वर HDMI भी है
Thank you, Mallikarjun. And it's a pleasure to be here. And at the foremost, I'd like to thank the organizers, Anju and Alok, old friends. And it's great to be here. And I think they've put a fantastic meeting. This is not counting in my time. Okay. No, no, I know Mallikarjun for a long time, so I know. Molecules which acts as processes and acids and actually recruits multiple cell types, dendritic cells, mast cells, macrophages, B cells, and T cells. But mycobacteria has several kits in its armory and it avoids the immune system, and we'll be talking about a few. So the pathogenesis, if I were to oversimplify it, mycobacteria, unlike other bacteria, are able to secrete many of its proteins, also called secretory proteins. And if anyone is familiar with the Monto test, which one actually goes through under the skin, it's nothing but culture filtrate. So before the sequencing of mycobacterial genome in 1996 by Stuart Skoll Group in Pasteur Institute, everybody was working with this like mycobacterial lysate and or let's say the purified proteins, using them for diagnostics, using them for as vaccine candidates, but of no use. And beyond the secretory antigen, some dead and living bacteria may also be released. Primary antigen presenting cells or dendritic cells go to the nearest lymph node, initiate CD4 and CD8 T cell responses. These T cells are then migrate back to the infection and help macrophage kill. This is the end of the Yes, true for most pathogens. Actually use that to show that like the first question that we are talking about through this session. I have also for pathogen physical proteins which responses. Terminologists like Anju would connect that for T cells, primary stimulatory responses, the threshold is right here, and T cells are here just to prevent autoimmunity. But then we thought that, okay, we go back to the drawing board. After the release of the secretory antigen, certain beta-living bacteria may also be there. If they are taken up, that means that they will be taken up by the same dendritic cells made by the antigen in the first stage. So when we did that, we found that the immune response completely reversed. An interferon gamma, high levels of interferon gamma, if you take the antigen disease without re-stimulation with TB, it gives you a beautiful Th1 protective response, high interferon gamma, low IL-10. But then if you do it with re-stimulating dead and living bacteria, the profile simply reverses. You have high IL-10 interferon gamma, which means a protective response simply becomes a suppressive response. There is no sterile immunity. And salmonella, plasmodium, these are times back. You will get infected for 1 week, 10 days, maximum outside of say, 3 to 4 weeks. And then you get it out of the system. It is extremely difficult. There is nothing. I'm sure all of us out there are infected, but they are healthy simply. Like which you are supposed to deal with. The calcium itself. Now, any T cell responses that are upregulated are insult. From there they go, they add these Trojan horses and it avoids killing Okay, so this is like a repetition. But what we found out was, in all these cases, calcium played a major role. And we found out that calcium is very important because any fibroblast it takes up, makes a fabrosome or a centrosome, fuses with the lysosome, which has the lytic enzymes, and then this fusion to occur. And the MHC molecules are there, the proteins are chopped, presented on the peptides, and then you get T cell responses. So, but this fusion to occur, 2 things are important. One, an intracellular concentration of calcium is very, very critical, And secondly, the pH of the fusion must be less than 5.5. It actually secretes urines, which alkalines the ribosome, and also secretes ammonia, which is again very basic in nature. So, and plus there are multiple molecules that are expressed. I will not go into the details for the short of time. This is important. For this, calcium is required. It also induces autophagy and apoptosis of macrophages. Calcium is actually very critical, but roots of calcium are very important. Calcium can come from various roots, from voltage-gated calcium channels, from the store-operated channels present in the ER, and what are known as calcium release activity channels. Where is it that the calcium is coming from? So that the channels were not channels and see what happens. So this is the and then so it is a classical reason using very nice actions. I this is so we thought that okay, so what happens in the patient? Same patient. And then we can see there are many alpha-calcium blockers that are used as antihypertensives. So we did experiment and then we found out that we have better. Also, now the child with this calcium blocker which take the system. So that was what it started off as
Scientifically, when you say by mistake you added the CFP and antigen in the cells, and then now we have a drug which hopefully will come out pretty soon. And we found out that today is going to make a point. I'll just quickly go through this one. Some of this stuff that is goes to the calcium channel, the toll-like receptor pathway, the high expression of this channel cell goes to the toll-like receptor pathway. You use SRMs to knock down the specific proteins in the DNA pathway, you will see your path expression. Okay, and then we also show that they can reverse. Process and take the different so we can calcium channels are also targets for cancer. In fact, I was wondering whether that is the reason why BCG actually works like this when you do the other cancer. It's becoming prognosis is 100%. Maybe it increases by engaging channels and then it does the reverse. And we find that our molecules as well the channels. This is something I wanted to be quite sure about. This was the first book where I published in 1996. So the big quality is a part of unusual. We showed that it's supposed to be and we showed that also at direct voltage gated sodium channels and calcium channels also, so ion channels basically. So this is where the nutraceutical component comes in my talk that we will looking that we can explore, and some of my this year's fourth semester project nursing college students have asked them to work on curcumin and CAPE to look at some of the TB responses to see whether it has any influence on the regulation of potassium channels. And maybe one student from zoology this year will join me for PhD. He's working on potassium channels. So calcium में बहुत कम हो गया, बोर हो गया, थोड़ा potassium किया जाए. So we thought that this is not true. The calcium homeostasis is not restricted to TB. It works as a liaison with TB, HIV co-infections for the shortage of time, and Staphylococcus also. It works in Staphylococcus also. That Staphylococcus aureus also is done in ICBR. All this is published. I just didn't want to put names. And also Streptococcus. You know, this is the expression of calcium homeostasis. That if you block intercellular calcium, you see PDL1, which is a immune checkpoint inhibitor used in cancer. This gets inhibited. Whereas if you use EECA, which blocks extracellular calcium, the response is the other way. So it's just not the calcium that we have identified. The root of calcium homeostasis plays a major role. And then we just recently published a paper in Immunology last year. It showed that it is also restricted in involving T cell homeostasis, T cell activation, T cell function, memory to activation phenotype of the T cells, and how the effector functions of these T cells also. That if you take, let us say, calcium activated T cells, or calcium, let's say, the TMDA, intracellular block and extracellular block T cells, add those to infected macrophages, you get a reverse phenotype. You see high doses here, if you block it one channel, one calcium channel, and the TB is clear. It's all GFP BCG or the GFP MTB, and here in one case it's clear. So depending on how calcium is rooted in these cells, it actually can have a devastating or a protective effect. And I think Mallikarjun
You've given me enough notes, so I will stop here. Right? So this is how the story. This is a we also have next year. Oh, sorry, next month. Oh, great. So we can take your questions. So this is the group in ICGAB. Shashank was the one who identified all the voltage gated channels. Mohit was the streptococcus and the David and data antigens and identified the DT. And this is the group in ACPR. Ankush is sitting here. He has a poster. He's looking at short chain fatty acids in regulation of tuberculosis because we want to get into non-metabolomics and other stuff in collaboration with gut biologists who I don't see here, Rajagopal Raman. And these are the people of the group. And I thank you all for your attention. Questions? Yeah, please.
Yeah, actually, see, all these NTP, antigens, whatever you are working, or maybe other people are working in India, all these the TLR pathways.
No, that's a very good question. So a couple of researchers show that some of these RGs, like CFP10, they bind to TLR2. It was a Nature Immunology paper, but unfortunately, not many researchers could replicate those studies. So it's might take a pinch of salt. So that's what I made about. You know, like this TB antigens have been worked for almost now 60-70 years.
I saw several papers actually from India where they're just throwing the peptide and losing the effect.
Yeah.
How they are doing, nobody actually knows that.
But if we show data to show that like, it may not directly bind to it, but it does influence TLR responses. I will tell you how I did for the slides. We identified using bioinformatics, we identified an antigen called 35296. Right? So this girl, Upasana, she did that. It has a strong homology to one of the death domains in the MID88 pathway. So we have another protein which has the class 6 domain. Right?
No, see.
No, so when we add this, it does not bind to TLR2. But what it does is that it goes and quenches all TLR pathway. It directly interacts with IRAK1 and thereby prevents IRAK1 and the MID88 pathway. It's like a sponge. It acts like a sponge. There are many antigens which do the same thing, but they don't seem to bind to any of these receptors.
You are telling actually it goes. They are actually he is catching up.
Yeah.
So how it goes?
It goes by 3 receptors. Toll-like, you asked about toll-like receptors, so I said
Whatever you are making like that, it's TLR4 something like that.
no, this is an antigen which blocks TLR2 or TLR4 signaling pathway. I said. Your question was how is it going inside? So there are 3 receptors that have been published. One is B6-9R1, CD209, which again one of my students is working. These are CTAC-lectin receptors. Just like your TLRs are toll-like receptors, these are CTAC-lectin receptors. These are ordinate receptors. It goes to mannose receptor and also goes to MR1. The MR1 is mannose receptor 1 and one more receptor, CD40. It goes to CD40. This was the earlier part. But then now they found out that the major receptor, this is a JEM paper in 2006. The DC sign, which is earlier known as GP120 binding protein because HIV GP120 binds to it. This actually goes and binds to TB and takes it. But that is not restricted to that. I mean, for example, look at Plasmodium. We have antigenic diversity there also. But not a single protein of Plasmodium is shown to bind to TLR2, but we have to be sure we can bind it. Right? So same questions come together. How does it go? Maybe it simply goes by simple phagocytosis. We don't know. But the responses and effects are there. People have been working, but then
can we also have said, can you make some of these recombinant protein, or are there any studies which suggest that these proteins are tagged with some chromosome or something? Where exactly and how exactly?
oh, we've done that, Anju. We've done that. In fact, when we right after our first paper, we did that. I spent almost 6 to 7 months looking at we knocked down TLR2, all the TLRs using siRNAs, it had no effect. So it means that then okay. We actually labeled it with PE, microenzyme 1 FITC, and then actually showed you by FACS, by confocal, by Z stacking, it doesn't work. All I know is that it goes inside. And if I block it, it doesn't. If I add, let us say, some of these diagrams like LPS or PAN, they don't seem to do anything to it. And I'm not the only person. This gets discussed in every TV meeting. That's the question that it gets discussed, but there is no answer.
Another question.
Yeah.
Because I finish then. So you are doing calcium channel increased by TB infection.
Yeah.
How is it?
So I showed them where there's a tail to pathway. I skipped those. Yeah. So, like I said, no, it is increasing where the toll-like receptor pathway. If you knock down, for example, this is increase, and if you knock down, it goes back.
Fine, this is fine. But see, how do you think? See, your protein is
How-
expressing more or something, or which transcription factors is, see, it is TLR4,
So,
whatever.
you mean the transcription factor? Oh,
How
yeah.
it is expressing protein, you are telling receptor level, that channel is increasing.
You mean the promoter activity of these channels is what you're talking about?
Either protein expression is more, or it is coming from other pool of the.
No, it is the protein expression. Oh, I think 28 is difficult to like to summarize 28 papers. No, on this thing. We looked at global and microarray levels. We looked at SAR in the libraries. We looked at all that papers. I have not talked about. I wanted to focus on that calcium thing. But we'll be happy to discuss it.
Yes,
Thank you for the time. Nice work. I appreciate it. Generally, inside calcium zones are their internal calcium stores. They are mostly like flux channels. Like in T cell, this was the report. So in this case of the hepatic cell, whether you find any correlation with this or after using BAPCA, did you use experiments?
Yeah. See, all the calcium channels and the calcium influx experiments are done with, we use fluorophore, we use ionomycin, we use some of these inhibitors that we use TMB8, EGTA. TMB8 is an IP3 receptor antagonist. It goes and binds to the IP3 receptor, thereby preventing intracellular calcium release. So that's the one. And then we use the voltage gated channel inhibitors and activators. Like there is a, if you it's amlodipine, which is the drug which is being like for which the clinical trials are going on. That is an anti-calcium channel blocker. AK8644 is a compound which activates those channels, which actually gives you the contrasting responses. We have done those experiments.
So do you think when people are taking this blood pressure tablets, that may correlate with the TB?
We did that correlation there. So the TB patients, whether like any of these TB patients have like a high blood pressure or this thing, that data was they said that we couldn't find many people who are on these drugs and came down with TB. My mother herself was admitted to a brain tumor, but then she had a lot of lung problems also. So they said that let us try for TB. I said she would not get it because she's been taking this amlodipine for 20 years. So then it turned out it could be a correlation. But then the trials are going on. I mean, we published this data, and then Lalita Ramakrishnan, who is Venki Ramakrishnan's sister, she also works at TB. She published a paper the very next year on another drug, verapamil. So it's a combined drug trial by Axiomaris. And this is part of like the drug repurposing that we talked about. Metformin does it. Anti-TB, anti-diabetic metformin does it. It's a huge story. In fact, metformin is also being. But then this is not the case. That is not that amlodipine or something. It will find a way to beat it also. So.
It was a wonderful talk. I compliment you. Thank you. You've done extensive work. Some basic query which just hit my mind, not directly related to your work, maybe just my ignorance. Macrophages are very heterogeneous population. Tissue specific variations are there. So does that impact their transformation into dendritic morphomes? Tissue variation, because alveolar macrophages are the main ones which could be studied. But there are other regions where TB takes place and macrophages are.
Yeah, so you know that there's also some students in macrophages, M1 versus M2, M1, M2A, M2B. So we have not looked into issues macrophages.
No, I just thought that maybe you are aware that somebody else,
I have not come across it. Most of it is with alveolar macrophages or TBMC differentiating macrophages. Extraordinary TB is there, but then what the theory is that it always a migration from the primary complex to TB will not go directly in infect. It has to be via so
The pathway that.
yeah. So what they say is that it is the alveolar macrophages which actually take it. Using technopacks and other story I can say
And can the percentage of genetic cells that are differentiated from the infected bacteria be used as markers for diagnosis? Percentage, say, a normal person may have this percentage of genetic cells.
window would be very low because the total population of this genetic cells is about 1%. So if you have 1% and then 0.1%, the population is very difficult.
Thank you very much. Excellent.
Thank you very much.
To felicitate Professor K. Natarajan for such an exciting book.
So she did a PhD from Chile. She was the she's
You, Selena. Nice. And Professor Natarajan has set the stage with the roots and pathogen interaction with mycobacterium as the pathogen and macrophage as the host. I will be speaking on another pathogen which is a eukaryote, and it also chooses macrophages its host, that is Leishmania. So we have similar kind of story, but a different kind of pathway. So I hope PH1, PH2, all the angles are there. So I tell you in a different way. So we have used pharmaceutical or nutraceutical, as you say, which has been taken since long in our Indian scenario, that is Gugulu. It's used for reducing cholesterol. So we found, in the literature that generally Leishmania uses cholesterol for infecting the macrophages. It starts taking that. So we thought, why not we use Gugulu and see whether it will, as an adjunct therapy, because many of the drugs have failed in the clinics and physicians are on the lookout for alternatives and there are no vaccines available. It is going out of hand. So visceral leishmaniasis, it's a vector-borne disease spread by sandfly, and the prevalence, it's mainly in the poverty-ridden areas of India, Northeast India, and it has even come to the northern eastern part of Uttar Pradesh. And visceral leishmaniasis is characterized by, fever. It's called as Kala Azar, that is the black fever. And if people are untreated, they develop, pigmentation. That is why Kala Azar. So it's found in almost 3 continents, as you can see here, and Muzaffarpur in Bihar in India is the epicenter of visceral leishmaniasis cases. And here you can see in India, these are the foci, and predominantly this is the area, hyperendemic area, and this is the Indo-Gangetic plains of Bihar, where we have hyperendemic places. So here you can see here, these are the different manifestations of the disease. And this is the most
it's causing a headache. It's a problematic issue for India now, that we have visceral cases decreasing and with an increase in the dermal cases, because now it has been hypothesized that the parasites, they are under the drug pressure, so they are escaping the viscera and going to the dermis. They are becoming dermatotropic from viscerotropic, and it's very disturbing to say that humans are the reservoirs. So sandfly will give direct load of parasite from man to man. That's a problem. And if they are present all over the skin, then the parasite bite is loaded with Leishmania parasites. So now Government of India and the WHO has been focusing on PKDL cases, how to reduce them. So this is the, as per CDC, sandfly host and the human host. There is a vicious cycle going on between these two. So what is our hypothesis based on that? Existing drugs have serious side effects, and there is a time when the parasites become resistant to these drugs. So there's a need for alternative drugs because there are no viable vaccines till now, and they are in the nascent stages. So we took the Gugulu, and as a control, we use statins because those are used as anti-cholesterol drugs, and which have given several good results on macrophage cell line as the host and the Leishmania strain. And here in the in our study, the main is we have used clinical isolate, that is the amphotericin B resistant, because amphotericin B is the only drug which is successful in the clinics now, which attacks the ergosterol in the membrane of the parasite. So we have chosen this amphotericin B, and this is our work plan. We have taken Guglu, it's a gum resin of Commiphora vitae, and we have, this is the way we done. First of all, we saw the hemolysis, because as per FDA, you have to check whether the drug is toxic or not. We used the hemolysis, human RBC assay, and PBMC, and we found out the IC50 doses, and then we looked for the direct cell killing. We have used two strategies, direct parasite killing and intra-macrophage clearance of the parasites by giving by treating the macrophages with the drugs, that is prophylaxis we call it, in vitro, and we infect the macrophages with the parasite and then treat with the drugs, so that we call it as therapeutic in the in vitro setup. So here you can see we did the chromatogram, and we found that it has a standard guglosterone, which is the predominant molecule, and this is the these are the results which shows that Gugul extract showed very less cytotoxicity, which was not significant to peripheral blood mononuclear cells, and hemolysis was very negligent. So it was safe to say that they are not toxic. Then this shows, and LDCI is the clinical isolate that we used, and these are some of the values for IC50, and AG83 is the WHO resistant WHO reference strain. And so we could see these are the IC50 values using atorvastatin. Then we checked by different pathways, Leishmania killing pathways using annexin and PI. And here you can see Gugul, as compared to the amphotericin B, it causes 18.88% apoptosis and 61, so which was quite significant as compared to the standard drug. Then this shows the with the AG83 comparison, and we also checked, because one of the method of killing is the generation of ROS in the parasites. And again, you can see here, if you compare this is the clinical isolate, which is already resistant to amphotericin B, but when we use Gugul, it showed almost more than the amphotericin B. And these are some of the parameters for parasite killing, that is the loss of mitochondrial membrane potential. Again, you can see the treatment of Gugul, how significant it was as compared to the amphotericin B. So we checked the growth arrest also, and here you can see Gugul also renders growth arrest to the Leishmania parasites. And for the reference strain, then we estimated the ergosterol in the membrane of the resistant isolate and the AG83. And you can see these are the estimates of the ergosterol content and how our treatment suppresses the ergosterol content if we treat with Google and amphotericin B or atorvastatin. And we did some of the SEM images because why we had to do, because the shape of the parasite is very necessary in order to enter the macrophages via the complement receptors. So they have to be spindle shaped with lot of flagellum movement. So here we can see here, this is the control and this is the treatment with Google and this one, Google, and you can see at IC50 values, it has made them puffed up and the flagella almost is lost, and IC90 values almost destroyed it. So conclusions that, that they were indeed, it is effective in direct cell killing. Then we focused on some of the proteomics using SWAT-based differential proteomics, using amphotericin B and Google. And this is the proteomics workflow for both AG83 and the clinical isolate. And we were able to identify about 818 proteins in AG83 and 1,944 proteins identified via the proteomics. And these are some of the workflow for annotation of uncharacterized proteins. One of the fallout was that we were able to find out several, because still the Leishmania donovani strain has not been fully characterized genome, whereas L major has been characterized, which is a causative agent for cutaneous leishmaniasis. So total number of proteins identified, and these are some of the upregulated and the downregulated proteins. And some of the, when we did gene ontogeny studies, we could find in the isolate that most of the proteins were related to molecular function and biological processes, which were, upregulated and downregulated. That was mainly on the molecular function. So now, we are trying to validate by using different assays. So these are some of the hub proteins using in the AG83 here, and in the clinical isolate, 19 hub proteins and 12 hub proteins were using amphotericin B and Google treatment. And when we did all the analysis, we could find that most of the proteins, they were ribosomal proteins, and enzymes for purine biosynthesis. They were significantly downregulated on treatment with Google. And we did some of the DEP pathways and cellular process analysis, and we tried to correlate the assays and the DEPs identified, and we could find that some of these pathways, they were inhibited by these things, and which are very important for its pathogenesis in the macrophages. And we did several annotations of the uncharacterized proteins in this clinical isolate, and we are trying to validate, because these are just predicted at the moment. And we also did some of the gene ontogeny, which was very different, because over the years the reference strains as given by the WHO, if you culture it more and more, they lose their virulence. So it was very relevant to use the clinical isolate and do the further processes. And we did several annotation, and we could find some of the fold chain changes in the proteins, and these are some of the functions attributed to these different proteins that were identified in our proteomics. And what are the conclusion? Main conclusion drawn out of this study, proteomics, was that upregulation of these antioxidant enzymes and ATP binding cassette superfamily and up proteins like ribosomal proteins, purine biosynthesis, these could be potential drug targets. This was one of the fallout of the study. So next we used macrophage, which is the main exclusive host of Leishmania parasite, and they are important in this thing, and they elicit an immune response or enhance, because TH1 and TH2 pathway. What we are looking here is TH1 pathway, which should be pro-inflammatory, and interferon gamma, especially, as you said. So somehow again, when the Leishmania enters the macrophages, it prevents the phagosome from meeting the lysosome. This is the story, and people are working out on that. So this is the workflow. We took THP1 monocyte, we differentiated into the macrophages by PMA, and we checked the cytotoxicity. Then we labeled here we used cyber green safe stain for labeling because unfortunately we lost the GFP labeled Leishmania parasite. So we chose this pathway, and this is an indicator because we could see the intramacrophage Leishmania parasites by flow cytometry. And we used DAPI stained and we used confocal imaging. And these are some of the cytotoxics shown, which was, and we chose the IC50 safe one. And we checked the ROS level in response because reactive oxygen species are one of the arsenals of macrophages for killing intracellularly. And we could see we did time kinetic studies, and we could see that this is the Google was very, very effective as, especially the 6 hours treatment was very effective in generating the ROS. And then we evaluated the nitrite levels also. And again, we found the same kind of response that it led to the production of high nitrite levels, which are leishmaniacidal. And then we established the macrophage and Leishmania infection ratios because those are very important for in vitro studies, 1:10. And even when we give when we use mouse models, we have to choose the right number of parasites to be given by the tail vein. So here you can see the confocal images of the macrophage infection. These are the cybergreens labeled parasites in the macrophages. And we could see the prophylactic effect that it was indeed rendering the macrophages leishmaniacidal when we treated the macrophages with Google and atorvastatin and amphotericin B. Here you can see that Google was very effective in keeping the infection in control. And again, the therapeutic also, there was significant effect as when we compared both prophylactic and therapeutic. Indeed, Google was leishmaniacidal. And then we compared the prophylactic and therapeutic effect. Here you can see Google, as compared to the control. We use cholesterol also because generally the parasite uses the cholesterol for making its own membrane in order to fight out the all the reactive oxygen or nitrogen species of the macrophages produced. So here you can see these are some of the. This is the control where there's no parasites are there. This is the control with macrophages infected with the parasite. You can see the L. donovani bodies. These are the LD bodies. And this is the where Google treated. We have the this one shows the cholesterol treated because when we treat with the cholesterol beta cyclodextrin, so it is not able to clear the parasites. So these are some of the we used, for Nile red stain for cholesterol, and we could find that it could not, because wherever you see Nile red, it is surrounding the amastigote ridden, the vacuoles full of amastigotes. So Nile red means it's an indicator that is trying to draw the cholesterol for its own good. So sum total, because I want to sum up because of the shortage of time. So our finding said that when we compared prophylactic and therapeutic potential, it pointed out to significant reduction in the intramacrophage parasite infection when we treated with amphotericin B, Google, or atorvastatin. And this also showed an increase in parasite load that indeed it proves that cholesterol is one of the pathways for the infectivity of macrophages. That's why taking Google can help. It can be an adjunct therapy while using Vidwin. So it can be used in combination. That's what our message to the physician communities. As we are in collaboration with Professor Shyam Sundar, who is one of the global heads of the Kala-Azar Research Laboratory in BHU and its field site in Muzaffarpur. So in future, because now there are a lot of cardiovascular problems which are attributed to increase in cholesterol, bad cholesterol, whatever. So people are taking this in, from Vaidyas and all that. Can it be used in a very specific way? So that's what our message to the community, physician community is. So I will give thanks to all my students, especially this is the PhD, one of the part of the PhD work of Dr. Chinmayi Bar, and I'm very thankful to all my collaborators, Dr. Mahesh J. Kulkarni, who helped us in doing our proteomic study from NCL, Dr. Sangeeta Samant, who really helped us in doing all the bioinformatics, because bioinformatics is out of, we are trying to learn bioinformatics slowly now. And Professor Shyam Sundar, who gives us all the strains, and we are in contact with him, like we are correlating with the status of the patient, how they are responding and all. Dr. Saha from NCCS, who is also a Leishmania biologist, and my funding agency, SERV and ICMR, and all my students who are behind this. Thank you so much.
Very nice. Just a couple of clarifications. One is using some of these drugs as drugs. It is better to use them as in combination with standard. Nice second and I was checking. That's certainly the standard. Typically, based on experience, talk about that when you're adding a new thing, always as a combination. It's not, at least not a lot of it. So, in fact, it may be advantageous to you simply because you can bring down the concentration of aerostatin as well as you can have
suboptimal time.
Absolutely. You may have to standardize, but then you can have lesser IC50s can be more, anti-50s can be lower. And since all these drugs have side effects, aerostatic side effects. Second part is that most of the work is ex vivo, that is being made at PhD1 or let's say mouse derived macrophages. On the problems, and I think the best example is curcumin. Curcumin works wonders in cell line systems. The moment you give it in vivo, the bioavailability becomes a major issue. When you're talking about, let's say, a Harvard drug like curcumin, then it has to be given for clinics. It will allow you to give it in province. Right? Or as an injection. So when you do Google what is the PKPD levels, those studies you may have to do. Best example would be to infect mice and then give these as plants. And I was taken as a tablet. That's what we did with our infect tablet. And we infected the guinea pigs and then gave them treatment. So we did a lot of standardizations. In fact, because we have to give a how much they take. So we measure how much water a guinea pig will take per day. So then adjusting the concentration in that water bottle and ensure that it goes. So for 10-15 days, we just measure to be good. So you may have to do a little bit of this, but very nice.
I just respond to that. Because this was one aspect of study. So later on, just as you said, suboptimal doses. So it can work effectively. So this will be done by other students.
Root of things.
Yes. And other things, we are Dr. Nasir Sahai, he has handled this.
I know.
Yeah, and a lot of lockdown. So we are working on that. Thank you so much. Yeah, you just spoke on much.
Taking on from there, most of the work is ex vivo, and parasite is using cholesterol. If cholesterol is there, it is not being clear, and there can never be no cholesterol in the in vivo system. So, what would be the efficacy of the treatment if I use it in vivo system? Will it be able to clear the
membrane cholesterol is always there. There's a balance between them.
So, how will
it is taken up by the spleen macrophages? There is a lot of pathway. It's not direct like that. It has to go via the skin when the sandpie bites. So it is taken up by these neutrophils, they die, then the macrophages are eat them, clear them, then it goes into the macrophages, goes into the lymph nodes, goes to the liver, bone marrow, spleen, there because that's why spleen, splenomegaly, and then dry that. So there's a huge complex targets. As you said, it's ex vivo, very simple to say this. What exactly happens? So, by using marker or fluorescein or imaging studies, really anything.
So we have a zebrafish model for this.
What image? Less you can have hamster, guinea pigs, hamster model, and very whether or not good animal models. So it's a very complex kind of answer. And we need sample by for taking natural reaction.
So, wonderful talk. I would like to ask one question. You work on one. So, those are the samples you have taken a culture on. What you have done is you have similar results when you have taken a clinical patient from patients?
clinical here, what is the highlight of the study is that this is the isolate from the clinical patient has relapsed to amphotericin B in the clinic. So that's the main part. And here, and the clinical data is showing different story, but when we are using in the in vitro, it is showing different story. The IC50 values are different.
And what happened to this like whole structure? Because it's a fragmented one. So where is really destruction taking? You see like 100% parasite is destroyed, but is it particular?
But not with amphotericin. And shape is very important.
Okay.
So that's why it was.
So shape is not changing at all. Yeah, when you have like high treatment of Google, it changes.
Why we are saying because when it is there in the circulation for a brief amount of time before going into the back. So if you take Google or any drug, it is under the influence. Will it damage that because it will prevent the influence?
Any other question? Yes, please.
After applying Google, have you checked the antioxidant pathway in antibodies? Is there?
Yeah, I am not sure. I have not shown gene expression studies. I have not shown TH1, TH2 pathways. So there are a lot of studies, but I just highlight them.
Because they have to pinch the cross production.
I'm not sure.
Basically, such an outstanding lecture.
Thanks, Dr.
Good morning and salutation to one and all present here. You know, many times I just write my name Sanjeev Kumar and my wife Professor Lalita is writing Lalita Gupta. So many times people call me Sanjeev Gupta. But today, somehow the gentleman called me Sanjeev Singh. Okay. So I'm really thankful to the organizers because they gave us a chance to visit Delhi University after a longer time and meeting the great personalities in science to discuss the work. So I'm going to summarize the work what we have done with drawing some conclusions. So we know that we are born in the world of microbes. They are present everywhere. They can survive in any situation or condition we cannot even think of. So in this situation, how we have to survive? So that was a point we thought of to find out the reasons and how this is possible. And one fact we already know that we are all born sterile. That before birth we don't have any microbes in the body, but as soon as we are out in this world, the microbes are encountering. Some of them are beneficial, some of them may not be. As you can see this picture, with the age, the microbes is increasing, and this is a data about the gut of a human being. So many of the bacteria or microbes, they enter through different pathways into the body and they house inside or outside. And we believe that we have to survive with them because this is the word meant for microbes, not for us. They came first, we came later on. So these are the different routes by which they can enter to our body, and many of them are called endogenous because they survive there. And they perform many functions. And one surprising note is that they are, especially if we talk about the gut, they are 10 times higher in number to the eukaryotic cell we carry in our body. So having such a huge load of microbes, how really our immune system manages? And getting the benefits from the microbes and not getting any harm. So it is a kind of very fine balance which is required. And especially when we talk about the gut, you know, if this fine balance is not maintained, we end up in some chronic, So this self-tolerance, what I talk about, means not harming to the self, rather harming to those microbes which are pathogenic or harmful to us. And in parallel, not killing those microbes which are beneficial to us. So let's think about it as a kind of very complex question itself, that how the system manages it. Because if we end up not managing this path or this mechanism, then we end up in some kind of inflammatory bowel diseases. That means the immune system is start reacting against the microbes which are present in the gut. And it is a devastating situation for someone's life, because we cannot survive with the microbes. And if we start reacting with the microbes, then there is a real problem starts. So we try to understand that this situation is named as inflammation energy. That means the immune system is there in the gut, but it is not under the process of activation. And the activation of immune system is also called inflammation. So energy is inactiveness or non-reactivity. So we try to find out the question of such type of questions in mosquito system, because I worked at NIH as well as in present laboratory I'm working on the mosquitoes, especially on some peroxidases in the gut and how they handle the situation with bacteria and plasmodium. So first of all, we thought to compare human and mosquito, and we found the mosquito and human both are similar in terms of housing the microbes. As you can see, in human, at different parts of the body, microbes are present. And in mosquito also, they are present on the body, they are present in the respiratory system, they are present inside the gut and so on. So we thought it can be a good model for us. And on the other hand, we all know that mosquito, they take blood. So first of all, we analyze that when they take blood, what happens to the microbes that are present in the gut. And we found with time, there is an increase in the number of microbes. So after blood feeding, the mosquito's gut was dissected at different time points, and the bacterial level was determined by using 16S rRNA levels. Now, in parallel, we found that there is a specialized peroxidase gene, and this is a secreted peroxidase, and this gene was also induced along with the time kinetics of blood feeding. So arrow, this triangle shows you that when this gene expression was very high, the level of bacteria was low. But when the gene expression was coming down and around the 24-hour, you see the bacterial level was high. So what we thought after seeing this result, that somehow this peroxidase is acting like a myeloperoxidase or eosinophil peroxidase, because they are known to be antibacterial. So with this question, we moved ahead to identify the role of this gene, and we thought to silence this gene. And the silencing was achieved by the double-stranded RNA-mediated RNA interference, and then we analyzed its effect on the bacterial growth or bacterial level in the mosquito gut. So we take 24 mosquitoes, one they were controls and one they were silenced. And we look for bacteria present in their gut. And what we found, that the level of bacteria was suppressed. But we were expecting that there should be an enhancement. Then we took the individual 24 mosquitoes and analyzed each individual gut, that how much microbial load they have. And now again we see this result remains same, that the microbial level is going down after silencing this gene. So that was a big surprise for us. That means this gene is somehow not only it is not an antibacterial, it is protecting the bacteria, or it is helping the bacteria to grow. So now question comes, in what way this gene is protecting the bacteria? So first of all, we thought as a normal question that antibacterial genes, let's look for their expression. So in this control are shown in blue color and silence are shown to you in pink color. So first of all, we look for how much silencing we can achieve. So you can see we have a pretty good silencing, we are about 80–85%. Then in those samples, we look for some of the genes which are called immune genes in mosquito. And you see all the immune genes are upregulated after silencing the HPX15, which is a heme peroxidase. I have already discussed about it. Now we thought to give antibiotics to the mosquito and doing the same experiment. And again, you see in the first pan and in the first left-hand side, blue color is giving you control and the pink color is giving us the results of the silenced one. And what we see again, the silencing is pretty good and there was no increase of these genes, neither in control, not in silence. That means the microbes were not present in the gut, that's why these genes were not induced. So we did many experiments, but because of the shortage of time, I came with a model, and what we found that this is the picture of gut, and gut epithelium is mentioned on the top. So gut epithelium is immunoreactive. As we have already said that this is an organ for digestion as well as for immunity. So making a balance between the two phenomenon, digestion or immunity, it is very important for every organism. Because the main purpose should be the digestion, not the reactivity. So what we see here that gut epitheliums were cells, what they do, they secrete mucins. And this mucin is cross-linked by tyrosine to tyrosine residues of one protein, another protein, tyrosine residue, and it makes dityrosine. And that dityrosine make a network over the epithelium. So directly the antigens present, the microbes present in the gut lumen, which are increased by blood feeding, they are not coming in direct connection or contact with the epithelium. So when there is no recognition, then there is no reactivity. But when we silence this gene, we found that this dityrosine network was broken. It was not developed at all. That means now the antigens are in directly connection or directly contact with the epithelium, and if they are recognized and immunity is induced. So this barrier concept was really well taken, well accepted by the scientific community, and this modulation, how it is really impact all organisms. So I try to explain that also here, that how we have to survive in the world of microbes. The one thing is that immune system, keep tackling with them, killing and removal. So while killing and removal, we produce many toxins like superoxide anions, nitrites and so on. So they may be harmful for the beneficial bacteria as well as for the host. Because we know chronic inflammation is caused by such kind of molecules. Or there may be another way that we have to make a cross-link barrier between the immune system and the microbes. So they do not see each other, they do not react with each other, and they live happily is a kind of policy, live and let live policy. So get the benefit from them, do not kill them. So this work was very well accepted in the scientific society, as I said before. Now, a report came from Imperial College London after 5–6 years of this work, and what they found that this peroxidase enzyme is also helping the sperms or male gametes in the mosquito. So before I come to this conclusion, I would like to show you here that how the reproductive system works in mosquito. As you can see that this is a female mosquito, and these are the ovaries. They have a common opening and to the outside. What happens then? Mating happens in these insects once in lifetime. And the sperms are stored in a specialized organ that is called spermatheca. Now, when this mosquito takes the blood, eggs are matured and they passes through the arrow which has been shown by the blue color. Now, sperms are passing through the arrow by the red color, and at this time, at this point, these eggs are fertilized by the sperms and they come out of the body and they develop into a new life. So I again repeat that only once in life there is mating, and these sperms are stored for weeks. So this group which I talk about, what they have found, that mating, no mating, or blood feeding, how this gene is really induced or regulated especially in the spermatheca, where sperms are stored. So let's compare this structure with the gut. In gut, we say microbes are there, gut epithelium is there, which is an immunoreactive. Here again, spermatheca also has a covering of the cells, and the sperms are stored in that lumen of that covering. So no mating, on the first layer you see that this gene was not induced, but when there was a mating, this gene was induced. And when there was a blood feeding after mating, this gene was highly induced. That means this is somehow playing a role with the viability of the sperms. Now what they have done it, this is not my work, this is their work. What they have done it, that in 2 experiments, they looked for the viability of the eggs that were shed by the females. So only it is a given fact that those eggs which are fertilized, they survive, but those eggs which are not fertilized, they do not survive at all. Because in the eggs there is a specialized phenomena happen, they undergo melanization after they are released from the body. In other words, when they come out of the body, they are white in color, but after half an hour, they all become black. That is all because of the melanization. So those which are not fertilized, they do not undergo melanization, so they do not survive in the nature. Now, what I see here, there are 2 experiments in control and silence. They silence this gene in spermatheca and look for the survival of the eggs. And what they found, that infertile eggs, as you can see on the left hand side, the percentage of infertile egg was higher in silenced mosquito. In 2 experiments, they have shown it, and there was a statistically significant difference. So now again, I thought, because they have not discussed these things in very extensive, they discussed my paper very well, or rather our paper. Dr. Lalita was also with me in that paper. They have mentioned, but they could not find that what was the reason that this was protected to the sperms in spermatheca. Now, I have a picture from some sources, and you see this is the structure of spermatheca, and this is the outer covering, which has many cells, including epithelial cells. And what you see in this reservoir, there are male gametes present. Now, cuticle is present here, and I found in some other references that this cuticle is having the same property, like it can undergo the cross-linking. So I believe this cross-linking is responsible for, again, making a barrier and inhibiting the interaction of male gametes with the immunoreactive epithelium. So, such kind of a compartment we think in our body, they are making, probably, this is my assumption, by making such kind of barriers, an immune zero environment. And this immune zero environment is responsible for the growth of microbes for the benefit of the organisms. And with these words, I really thanks to the organization one more time, all the audiences which are listening here, and funding agencies, BITS Pilani, because I worked before at BITS Pilani, and Carolina Barlas Muri, along with her we worked at NIH, and Madam Lalita, she's sitting here. One of our students, he has been acknowledged here, and Google, scientific media, and all platforms where from I got this video. Thank you very much. Any questions, please? This is important to for Google nowadays.
We had a different Google.
Thank you so much. Now
My pleasure. Yes.
open for questions. Yes, we'll start with Monica.
So again, this might be me being ignorant. I work with fruit flies and so in fruit flies often after mating or after the kind of nutrition there is a gut growth and size of the gut basically increases. So the change in the microbiota, do you think is it related with what first question, are there any changes in the size of the gut upon blood feeding or on mating in mosquitoes? And whether the increase in the microbiota that led that to see, is it related to that increase in the gut growth as
Well, there is no direct correlation we found, but we have found that mosquito can take almost 6 times larger blood volume to its own weight. So extension, that's why so engorged. So after you just allow mosquito to feed on the skin and you see they engorge like anything. And then you hit them, it is really pleasurable movement, because they cannot fly. So gut microbiota really changes after the feeding behavior because in Drosophila, they are never blood feed animals. So they have mostly the fermenting bacteria for rotten food. But in case of mosquito, they have bacteria which can lyse the blood. So hemolytic bacteria are there, which flower changes, of course. But with mating, gut, I am answerless here. Really, there is a correlation. I believe because they are both different organs, so probably there is no direct correlation. Sir.
So the microbes in both must be very different. Mosquito and human. Is the mucus layer very similar in both?
We looked for Drosophila, and in Drosophila we did not find any such a strong mucus layer, but there is a mucus layer which is always found after the blood is taken. I tell you one more thing in addition because you ask a very specific question here. Like when the blood is taken, there are some pre-made proteins. They are all thrown into the gut, and what they do, they make a matrix around this blood which is called peritrophic matrix. And after that, the mucin is secreted, and that make a cross-link barrier. So there are two barriers in mosquito. In Drosophila, no two barrier concept was there. Now we are going to ask a question here that why there are two barriers. So one barrier has a larger pore, the peritrophic matrix, and another barrier has a very small pore that can allow only the digestive enzymes to come to the gut. Not even bacterial initiators are allowed to enter into the gut because their pore size is small. So Drosophila situation is different because they never take blood. They always take the rotten food. Peritrophic matrix is there, you're right.
See, in human, there are segmented filamentous bacteria. Then our immune cells are there, which are induced by these microbes. And they secrete IgA and other molecules to kill them. That's how they control the bacteria cannot enter into the. So I was wondering whether mosquito also has similar kind of situation as we have.
Like in our gut, we have M cells, which are immune in nature, and M cells, membranous cells. In mosquito are there cells which do not have microvilli, and they are different from the normal cells, and they are similar to M cells. And some reports I read, not my work, and they say that when bacteria is not present, then this mosquito cannot really handle very well with plasmodium. So bacteria again is required to enhance the immunity in such a way like human is doing. I'm sorry? Wolbachia, yeah. Wolbachia was introduced to the salivary gland system, in fact. Because the plasmodium at last ended in the salivary glands. I'm sorry? No. In Aedes it was done. In Anopheles also, some experiments were done. But it was introduced to the salivary glands, not in the gut.
So what I have understood, sir, can I just
Yes.
So what? So is this change is a transient change whenever the blood meal is being taken, the whole system changes in order to. So it's like it's a dynamic change which is happening with feeding and otherwise.
Yes, answer is yes. It is a transient change which happens because we try to look for any such kind of cross-link barrier in when there is no blood feeding, because generally the mosquito feed on sugar, and in sugar fed it was hard for us to look for or find such kind of structures because the bacterial load is very low. That may be one of the reason. And in addition, we found some of the immunomolecules are all the time turned on. So they control the population of the bacteria at that time specifically. But when there is a blood feeding, the bacterial level is sky high. And that only happens after this peritrophic matrix formation and the cross-linking happens. Because before that, again, those primarily immune molecules are keep on killing them.
So the number may change, but the consortium will be similar.
No, sir. The composition changes. The microbial composition.
For changing composition, there has to be influx from somewhere. So I assume that they are there. Their number will change depending on who. And when the blood is there, they may flourish or they may go down. But the consortium will be.
Yeah, this work they have done. Dr. Lalita has done this work, and they found there is a huge variation in the species of bacteria after the blood meal. As we human, we also know the kind of blood, the food we are taking, the gut flora also changes. And that controls our physiology and psychology and many things. You know?
The change in number, not species.
No, in the species also.
There is a species in that, but it's normally food, yes.
Also changes. Like humanity, bacteria number goes very high after blood meal. But before they are in tiny amount.
No, so that's what we are saying. Number will change, but not the composition.
Degrading bacteria, they are almost negative.
That's what I said. That's what
Right. No. So new species are less likely to. Mosquito.
Female mosquito.
Because males are generally neglected in mosquito. Yeah. So it's a mind, but no, this is true that female and male, they have almost similar kind of microbes when they are sugar fed, because they generally take nectar from the and in laboratory we give them sucrose. So general microbiota is similar, but some of the specialized microbiota are present which are hemolytic in a low amount, but their amount increases as I told before.
Does the dynamics of the bacterial load change for evolution?
No, that is true that in the female, it is very well reported that there is a change in microbiota after the food is changed. And in males, generally, if you keep restricting them in the laboratory, and another thing is they do not survive for a longer time. They die in 2 or 3 days. Female survives for a longer time. So less likely there is a change because the diet is restricted in laboratory. As well as outside, they take nectar. If they change the nectar from one flower to another flower, then they might encounter different kind of microbes. This is my assumption.
So
You know, human is the most notorious animal born on this earth. I'm saying because I'm human. Because the kind of physiology, the food behavior we have changed, our physiology has not been changed in accordingly. You take the example, बहुत गवार वाला example है. गाय को लीजिए 10,000 साल पहले भी घास खाती थी, आज भी घास खाती है. लेकिन इंसान ने अपने आप को कितना चेंज किया. How much we have changed our food behavior. You can see last 20 or 25 years, nobody knows what is pizza, what is pasta, what is macaroni. But now, kids want only those foods, no other food. Roti chapati तो उनके लिए गायब हो गई है.
But we still eat chapati and
But not I have observed my son. Give him pizza whole day, whole month, he's happy. No, but my thing to say is that the food behavior is changing, and that's why the microbial community is also changing. And you know, then again, on the top, different diseases, they also have a change in the microbial flora in the gut and other parts of the body.
so much, sir.
Iron knockout in larval stages or in adult stages?
In adult stages.
So, what would be the difference if it had been formed in larval stages? Because sometimes we get that stage, the mosquito acquires bacteria and the bacterial environment is starting to develop. So, there will be a difference.
You know, when we talk about metamorphosis, there is a huge change in the gut of the mosquito. And it has been found that mostly the bacteria which are present in the larval stage, they are almost negligible in the next stage. So there is a complete transformation. But it's possible. But now this thing is like how much this, how longer this silencing will remain a factor. That is another question. You can see again, you do parents, kids are born with the silenced. But in Drosophila and mosquito, it is not known for that. Thank you very much. One more time. Thank you.
Now I would request Professor Mallikarjun.
How many times you have been speaker and Dr. Gupta has been chair?
So, last speaker, I think Shaida is not here. So I would like to invite Dr. Sabasati Secretary, Assistant Professor in the Department of Human Genetics. University of Punjab. He's here. So, genetics engineering week of primary diseases, students major from Delhi University and visiting PhD. So I would like to invite Professor Sabasati.
Thank you, Professor Gupta, for a kind introduction. So I'm very happy to present here. And I'm thankful to ICD to invite to me in this conference to deliver a short talk on the work that we have done in last 3 to 4 years. My group is very young. I have just joined in 6 years back. And this group is working on genetics of celiac disease, which is an autoimmune disease of small intestine, duodenum. And the main effect is basically the venous atrophy and induced by a protein called as gluten that gives a glue kind of property of weight, weight and body. and disease acts as a the principal antigen. And what it does actually in celiac disease, they increase the transmembrane permeability that causes the activation of T cells driven by TNF alpha. And the autoantigen is transglutaminase. So the main problem in these kind of condition is that they have several extraintestinal manifestations, including infectious disease like TB or different GI infection, and also lymphoma. So can we have, can we predict this disease? Can we have specific therapeutic targets? Because as the last speaker, Professor Sanjeev was talking about, the gut is full of microbiota, and there are many different kind of competition happens throughout the lifetime, and people may get different infection because of increased transmembrane permeability. So when we are talking about different extraintestinal manifestations, we did a microarray study, and we looked at more than 200 genetic loci to find out genes which are implicated in celiac disease. I will not go through those graphs, just summarizing my finding, our finding that in juvenile and adult celiac disease, why we segregated juvenile, childhood, because childhood celiac disease has much more of genetic component. Because CD is a complex disease where genetic environment, genetic component, and environmental players play a role together. So in childhood genetic, they have a much more genetic component rather than adult celiac disease. But when you look into the data, we found, don't go to these statistical values, we found that ANK3, ankyrin 3, that codes ankyrin G protein, has come up in a very good way. Look at the significance level, -7, -5, and it's associated with the disease condition. So then the question is, this gene is known for neuropsychiatric condition for last 2 or 3 decades. And their role in neuropsychiatric conditions are very well explained in case of bipolar disease, schizophrenia, anxiety, major depressive disorders, and several other kind of conditions where ANK3 is very much implicated. So when we find out this gene in celiac disease, like an autoimmune disease condition, first we wanted to look at how this axis is working, how this particular protein, which is actually present in myelin cell and implicated in different psychiatric condition, how it plays role in celiac disease. So are those individuals who are having neuropsychiatric conditions are prone to celiac disease, or individuals who are having celiac disease are prone to neuropsychiatric conditions? So this question, and we start answering this thing. When you look into the published reports, we found that there are several reports that say that there are psychiatric conditions are one of the comorbid conditions in celiac disease. And in our dataset, we have found more than 30% subjects having celiac disease are developing neuropsychiatric conditions over the time. And then we've gone ahead with our data. We did a meta-analysis with existing reports, and just look at these P values, that we have found that these are the 6 different neuropsychiatric conditions which are already published, and with our data, we found that they are showing significant co-occurrence among celiac disease patients. So, what is the molecule and how these 2 diseases actually co-occur? Can we have a specific drugs? Because for autoimmune disease there are no specific drugs. Mainly we target T cells, mainly we target to suppress the immune cells down. We give anti-inflammatory drugs like steroidal drugs. There are no specific drugs available. So can we find out a target that can handle both these disease conditions? Then we looked into the published papers, our own data. We found these are the 14 genetic loci across the genome. So there are not only one ANK3 that we have found. These are the 14 different loci which are common between neuropsychiatric conditions, schizophrenia, bipolar, anxiety, and depression. And they are common with celiac disease. So these are the chromosomes, genomic location, and the genes. Mind you, this HLA-DQ A1 and DQ-B1 are principal diagnostic markers for celiac disease as well. So when we diagnose celiac disease, doctor asks for HLA-DQ type. So HLA-DQ2, HLA-DQ 2.5, and HLA-DQ 8. they comprises almost 95% of celiac disease patients. So if there are 100 patients, 95% are positive for HLA-A1 and HLA-B1. And surprisingly, these two genes are also found in these 4 conditions. And our target gene, ankyrin G, that codes for ankyrin G, are also now shared between these 2 disease conditions. This thing is basically present in schizophrenia and bipolar disease. So these 2 locus actually gives antigen presentation and adherence junction. Then to find out what are the other genes, how they're in pathways and etc., we did a transcriptome study. So in our discovery cohort, we had a very small sample size, 3 CD patients and 4 non-CD controls. So these are not healthy controls. They have certain issues. So that's why they go to the doctor. They perform biopsies and we got the sample in the ethical permission. So those controls are basically suffering from dyspepsia. So those are not immune disorders, not infectious disease. Those are simply gastric ulceration. So these controls are not completely without inflammation, but obviously they are not, as depressive like celiac disease. So and these 15 and 21 green colored numbers are basically replication cohort, but we tested using real-time PCR. So we found that 135 genes are downregulated and 788 genes are upregulated. I'm not presenting the whole data, but for ankyrin G and CTH, that is E-cadherin, they also came up as a very significant gene which are downregulated in celiac disease patients. So what we are suspecting of the genetic study, literature search, meta-analysis, we found that in our study, these 2 genes are downregulated. And they are also implicated in multiple pathways. I'm not telling you all the stories, but these 2 pathways like mucosal membrane channels and synaptic membrane maintenance, these 2 are the most top 2 significant pathways that has come up in the pathway study using GATE and GEO, gene ontology databases. So that's fine. Then we had a biopsy sample size from DMC Ludhiana. Then we did for immunohistochemistry with the patient samples and non-CD controls. For this IAC, we used 15 CD patients and 12 non-CD controls. We stained them for ankyrin G and E-cadherin and beta-spectrin. They are actually interlinking 3 cytoskeletal components. So ankyrin G and beta-spectrin are cellular component, and E-cadherin know that it's extracellular and transmembrane protein. So we found that in all the samples, 12 and 15, though the staining patterns are a little bit different because it's a complex disease, not all the genes are always involved. But when we found out a pattern that there is a significant downregulation of ankyrin G in the ISC samples. These are normal EG stain, just to show the degradation of microVNS structure. We did not find any significant change in the pattern and the intensity of beta spectrum. When we did real-time PCR evaluation on 25 and 23 samples, we found significant variation in ankyrin G. But for E-cadherin, the p-value was not significant, maybe the sample size, maybe the heterogeneity in the patient samples. But for ankyrin G, we have found significant results. That indeed there is a significant variation. All of those samples was also included in the transcriptome study. Then we went ahead to find out what happens if we downregulate ANKG. What happens in the gut tissue? So we used IEC6 cell line and we downregulated the ANKG with shRNA construct, and we gave 2 induction to actually mimic celiac disease-like condition in vitro. One is pepsin digested gliding, that's the principal antigen for celiac disease, and TNF alpha, which is a pro-inflammatory molecule, also implicated in celiac disease. We standardized these 2 treatment doses after NTT assay. I'm not showing the graph here. So when we did this study, so this first graph is untreated control. Then we gave upper panel, I think it's not visible clearly. This is ankyrin, E-cadherin, beta-spectrin. So these are the 3 ankyrin, E-cadherin, beta-spectrin. This is for mRNA expression level, and we found that when we give these PT glidin and TNF alpha, all the gene expression comes down, and this come down is significant. This downregulation is significant. But when we give TNF alpha, upper panel is for glidin. When we give glidin, the downregulation of antigen and E-cadherin is quite significant. So if you compare these two graphs, you can see there is a significant downregulation. But for beta spectrin, the downregulation is not significant. The p-value is not significant. Same thing happens for TNF alpha as well. This is the Western blot. You can see the fed bands of NKG and E-cadherin in different treatment conditions. This is control, untreated, untransfected. This is knockdown plus glidin. This is untransfected plus glidin. Knockdown plus TNF alpha, untransfected and TNF alpha treatment. And this is the densitometric data to show the level of significance. So basically what we found is that PT cadherin and TNF alpha both downregulate NKG and E-cadherin, not beta spectrin. But when we downregulate, when we silence NKG, the downregulation is significant. So that means individual subjects who are having a mutation in the promoter sequence that leads to downregulation of NKG are more susceptible to react against glidin and application of TNF alpha. Now glidin is antigen for CD, and TNF alpha application can happen due to any infection condition or any chronic disease condition in the gut, maybe different microbiota interactions. So the subject who are having NKG downregulation, they are more susceptible. Then we tested our model for transepithelial electoral resistance, TAR values. We found that in NKG knockdown, there is a significant down of TAR. And then when we treated untransfected, transfected, it is transfected one, we found that in all the cases when we added TNF alpha and glidin, the TAR value is increasing significantly. So when there is a downregulation, that means membrane barrier is now disrupted. So if you remove NKG, the membrane barrier is getting disrupted. Second, we did the study with Dexter and FITC assay with 4 kDa molecular weight to check the transmembrane permeability. That means if there are two epithelial cells, what is the permeability between the two cells? Then we found out that with treatment, the permeability is increasing. Though these values not significant. These values not significant. Upper value are significant. Then we have looked into the cellular morphology, normal light microscope phase contrast. We found that glidin and TNF alpha is changing the morphology of the cell, but in case of antigen knockdown cells, the morphology change is very, very drastic. Just to look deeply into the data, then we did immunostudy, microscopy, and this is simple NK and E-cadherin in untransfected cells. Because you have seen that in untransfected cells as well, they are downregulating NKG and E-cadherin. And we found that the distribution pattern of antigen is changing when treated with glidin and TNF alpha. There is more accumulation of the protein on the nucleus. Then for E-cadherin, there is a breakdown of cellular membrane. I can't say breakdown, but obviously mislocalization. The localization of E-cadherin is now changing. It is coming more towards the nucleus from the periphery of the cell. Then we did this confocal study. These are the untransfected cells, these are the transfected cells. You can see the top picture where cells are untransfected, and the picture is showing a very intact E-cadherin structure and good cellular morphology. But when you look to the bottom panel, which are transfected cells where NKG is downregulated, you can see the cellular morphology is changing, and also the mislocalization of E-cadherin due to knockdown and the treatment of glidin and TNF alpha is causing the mislocalization that is changing the cellular structure. So that says that the function of antigen gene in myelin cell is well established. well known. But for celiac disease, it's autoimmune disease. Here, the function was not known clearly. So, a person who was suffering or having a lower expression of ankyrin gene due to any genetic polymorphism, that we have also found out genetic polymorphism for ankyrin gene, which is causing downregulation of the gene, and this may happen in the gut. Mislocalization of E-cadherin from the membrane, and most probably, we are not sure, we will be doing the study, that they are actually transporting those E-cadherin near to the nucleus. Maybe. So that's how this is the scenario that happens. So maybe this E-cadherin and ankyrin is basically connecting the gut and brain axis, where many, there are many studies, even Dr. Sanjeev was saying that, patients, that there are neuropsychiatric manifestations. Well, so just summarizing this thing, this is the ankyrin, this is beta statement, this is E-cadherin, that's the, membrane structure. This is the total protein of ankyrin gene, and this is how we are proposing the thing may be happening, that due to lower expression of ankyrin gene, the internalization or the mislocalization of E-cadherin that happens, and with the induction of TNF-alpha and beta glycan protein, there may be disruption of cellular membranes, and that may lead to celiac disease condition. So that's the first in vitro validation of non-immune genes in celiac disease. Other studies have used immune gene function to see how immune disease happens. But here we have presented a work where non-immune cytoskeletal gene can promote immune disease condition in genetically susceptible individuals. That means patient having HLA-A2 positive. I would like to thank Professor Tillman and Professor Sisler for the, facilitating us for this internship study. We are part of International Celiac Disease Consortium, and then Dr. Ajit Sood and Vandana Mitra for providing us a sample from Ludhiana, Dr. Siva and Dr. Somesh to help us in cloning and transmembrane assay, and funding support from SAR, DST Fixed and Central University of Punjab, and these are my co-students. Just previous week, my first year student, just presented this one because most of the work is from her PhD thesis. She just given the 3-page seminar, and most of the slides are taken from her, and they are my next 3 students who are working on celiac disease and vasculitis. Thank you.
What is the clinical relevance of ankyrin gene? It's
yes, it is downregulated. So, like for celiac disease, phenotype, when doctors are saying that there are many celiac disease patients where they are not confirmed by biopsy, because biopsy can see there is a degraded villi structure. But in present time, the phenotypes are a bit different. There is intact villi structure, but patients are having high TTP level and HLA positive. So they are searching for different other molecules that may be used as a diagnostic marker. And but if we look into the ANKG and how they are destabilizing the membrane, we can synthesis or we can propose any kind of new drugs or repurposing of drugs that may stabilize this process.
What is that the information which regulates the NKG?
Okay, so we are not into this thing at this point of time. This is a recent finding, just now. So we are now trying to understand how they actually mislocalize ANK3, E-cadherin. Do ANK3 directly do that, or there is any other partner that is helping in internalization of E-cadherin? Maybe that will give this answer.
And is the polymorphism responsible for the development of certain people? So is there same polymorphism in every patient, or do you find multiple cases?
So these are single nucleotide polymorphisms, and these are the odds ratio. This is very high. Odds ratio generally more than 1 is very high in number. So we found that these two SNPs are strong LDV promoter, linkage. So they have a promoter polymorphism, and these two polymorphisms are intronic. but they are in LD with SLP which are in promoter sequence. And when we look back into the eQTL, expression quantitative trait locus, we found that this polymorphism is altering the expression of ANK3. So these are the two polymorphisms that we have identified, but these two are not causal. Causal may be the promoter one, which is downregulated by ANK3. Right.
Very nice work. When you talk about E-candidate, at the time of junction between epithelial, there are many other junction proteins. So is E-candidate alone is that enough to have a breakage in that, or others are also downregulated or affected?
So that maybe can be answered by this particular slide, because there are tight junction, gap junctions, and adherent junction. Three types of junctions are present between two columnar epithelial cells in the small intestine. So when we knock down ANK3 and downregulation of E-cadherin happens, we can find there is a change in transepithelial voltage, right? But there is no significant difference in dextran FITC acid. It's a 4 kb molecule that passes through the membrane. So we can see there is no significant value because there are maybe gap junctions and other junctions that is still helping them to get the cells little bit closer. So that's why these molecules are not passing through the membrane. So we found this is significant because this is altering the charge, but not this one. We are suspecting that charge will be altered because in neuronal cells as well, because of loss of NKG from the myelin cell, the neuronal transmission does not happen properly. That's the biology. So we are expecting this thing to be coming positive, but surprisingly we did not find any good result in this assay. So that says whatever you are saying, that this alone is not able to destabilize, but obviously that will allow maybe the infection to happen. That because gut microflora influence in celiac disease is very, very well known. There are many studies that are found out that microbial populations are involved.
Size of gluten is smaller than this dextran FITC?
Size of gluten, because there are heterogeneous population. I mean to say type of gluten. So we say like 22 mark, 33 mark, 23 mark. So there are multiple sizes of gluten molecule that trespass those membrane barrier. So depending on with what size, because the smallest size is 10 or 9 mark. 9 mark. Okay, so all of them are having an immunogenic portion of gluten that induces HLA through T cell receptor. So yes, that's it.
Okay, thank you, Dr. Sabyasachi. Nice talk.
Professor Lalita for his enlightening lecture.
I am
Now I request to felicitate Professor Lalita for this session. Now I request Professor K. Natarajan to felicitate Professor for holding a chair for the session.